Hi, welcome to Science Hangout from Science Mission.
Hi.
Today we have Today we have Dr. Nan Zhang, who is the project lead on working on investigating zinc regulation during fertilization in mammalian oocytes in Dr. Woodruff's lab from the Northwestern University. Dr. Zhang has won several awards and honors to his credit. In 2003, he won Excellent Student from China Agricultural University. From 2002 to 2005, Dr. Zhang won scholarship from Chinese Agricultural University. He also won Excellence in Science Award from American Association for the Advancement of Sciences in 2012. Dr. Zhang also has several years of teaching experience. So he taught courses and was a teaching assistant in the University of Massachusetts and currently at the Northwestern University. Dr. Zhang is also reviewer for scientific journals and conferences. And he also has published several articles in peer-reviewed journals. So welcome, Dr. Zhang, to Science Hangout.
Hi, Sadashiva. Thank you very much for having me here. It's a great honor to be able to present my work in Science Mission, which is a pretty successful website and attracts thousands of attentions online. It's my great pleasure here.
Can you please talk about your background, your education, etc.?
Sure. Right now, I'm a postdoctoral fellow working in Dr. Woodruff lab at Northwestern University. I got my bachelor degree in veterinary science medicine from China Agricultural University at the year of 2006. Then I went to United States and obtained my PhD degree in animal biotechnology and biomedical science from University of Massachusetts at Amherst, the lab of Dr. Rafael Fisori at the year of 2012.
Okay.
So far, I have joined in Dr. Woodruff lab for 2 years. Now I am mainly studying zinc signaling in mouse eggs during maturation and fertilization in the project. So I'm trying to translate the zinc sparks technology from mouse to human and investigating gametes dynamics with the goal of improving fertility outcomes in human medicines in the near future.
Okay. And what motivates you every day?
Everybody wants to be a hero, and we know that the world is not perfect. As a matter of fact, it's full of problems. We are plagued by wars, diseases, poverties, etc. So the moment I learned this fact when I was a kid, I was determined to do something that can help our community get better. So I found my niche in reproductive science, and that is why I always ask myself a question. When I do science, can I apply it or not? When I choose or design a scientific project, what is the applicability aspect of this? So science, satisfies me because it can be applied to any sector, both in academia and in the industry. Another more straightforward answer is my strong curiosity. I just, love to be surprised by science. 99% of the time is that I have been motivated by my curiosity to understand how life begins. When I conduct research, I feel like I'm an adventurer on a journey of discovery, and that fuels my motivation every day.
Okay. And what are the challenges you face every day?
Currently, the research environment is very competitive, and I think everybody who works in the field can kind of feel that in the past decade. And nowadays the situation is a PhD degree is not a ticket anymore to academia career anymore as it once was. Therefore, the challenges that I face every day is my own growth. In order to successfully launch my career as a faculty, I need to produce a solid set of publications. I need to build up my network to find opportunities, and often I also need to try my best to search and secure my own research funding. So despite these challenges, I learned, along the way, the precious thing that I learned is to stay positive even when things don't work. I try to stay productive and keep producing novel data every day, because I believe in Albert Einstein's words, genius is 1% talent and 99% hard work.
Yep. And I know you have been working hard, and as a postdoc project lead, you need to put in lots of effort in getting the publication, writing, trying to write grants, doing experiments, everything you need to do. So if you get some time, what else you would do? Like, what are your hobbies?
I mean, first of all, I like to say my biggest hobbies right now, is hanging out with my daughter when I have time. That's what I'm trying to do. I'm trying very hard to try to balance my life work time and with my family. When I stay at home, I also love reading, video gaming, watching movies, listening to music. And I also enjoy cooking. Recently, I'm trying to brew kombucha at home. I don't know if you like kombucha or not.
No, I haven't tasted yet.
Yeah, I found it very amusing to brew kombucha myself.
Oh, okay.
When I go outside, I love photography, kite flying, thanks to my daughter. Skiing, swimming. Yeah, I love all different kinds of sports too. The only thing that I really love to try, but I haven't have a chance to try, is camping. My wife is not the camping person, so trying to convince my wife is the next goal I'm going to achieve.
Okay. And you mentioned about photography and all. So do you have any nice science-related pictures or which you can upload on sciencemission.com?
Sure. I'm glad to do that. Yeah, after the interview, I'm gonna look through my photo library and upload some beautiful pictures there.
Okay, that's good. Thank you. Let's talk about your Zinc project. Like, how did it come about?
Okay, so the Zinc project is actually a result of a collaborative work between a biological scientist, Teresa Woodruff, and her husband, who is also a chemist scientist, Dr. Thomas Halloran. So they initiated the Zinc project years ago, and there had been nothing before this work to suggest that Zinc would have had this important role in the development of a mature egg, and then in the transition of that egg into an embryo. Under Professor Woodruff and Halloran's supervision, I and the other two leading scientists in this project, who is Francesca Duncan and Emily Kay, decided to take this project to the next phase, which is to develop this Zinc technology as a non-invasive biomarker of oocytes and embryo quality. So fortunately, our work has resulted in many important discoveries on the non-invasive test of eggs' developmental potential. Our two most recent publications in scientific reports triggered the media's enthusiastic response recently. So I feel it's a great honor to present some of the data today.
Sure, go ahead, please.
Shall we start the presentation right now?
Yeah, sure. Go ahead.
Okay. Okay. Now you can see the slides, right?
Yeah.
Okay, just want to confirm. So first of all, again, I'd like to thank you for having me here today. I'm going to mainly talk about the Zinc project, which is one of the many exciting projects that are going on currently in Dr. Teresa Woodruff's lab. So most of the data I'm going to show you today are recently published in scientific reports. And the title of my today's talk is Zinc Fireworks, Review Egg and Embryo Quality. As a scientist, one of the challenges that I meet every day is to come up with great novel ideas. There are many ways to get great ideas, as you may know. Reading literatures, attending seminars, brainstorming, or have an intense discussion with your colleagues. But the thing that I want to emphasize here is that we also need to keep in mind that the involvement of technology and the development of science, they have always been a true two-way traffic, and we can always get inspired by focusing on the applicability aspect of science, by knowing what does current knowledge really needs from the scientist recently. Let me give you one example that is relevant to my research. excuse me, IVF and its various modified protocols, totally referred to as assisted reproductive technology, or briefed as ART. This technology is one of the most successful cases where science's applicability is maximally fulfilled. And until now, ART has been used in the birth of more than 5 million individuals worldwide.
Mm-hmm.
That's why to recognize this great technology, Nobel Prize was awarded to Dr. Robert G. Edwards at the year of 2010 for the development of in vitro fertilization. Okay. However, despite the importance of this technology, we need to keep in mind that this technology is not a perfect one. There is a lot still waiting for us to do to improve this technology for the goodness of the patients. For example, based on the ART National Summary Report at the year of 2013, which is the best resource so far to learn about current situation of ART, you can see that the average birth rate here in United States is only approximately 35%.
Okay.
Based on the bar graph chart. Right? So while this low successful rate is partially due to the nature of different diseases that lead to infertility, it also reflects the inadequacies of this technology. Believe it or not, let me right now dig deeper and show you what do fertility doctors usually do to fight with the low pregnancy rate and successful rate nowadays. They do majorly, they do two things. First of all, they do multiple embryo transfer, okay, which is shown here on the left. And second of all, sorry, I'm just trying to find my cursor. Okay. Second of all, they do extensive in vitro embryo culture before they transplant these embryos back to the mothers. And let me go to the data here specifically. The bar graph here, the pie chart here on the left shows that, you know, single embryo transfer here only accounts for 20% of the cases here, and the rest of them here, and you can see like 76% of the cases were performed with multiple embryo transfers. Now let's take a look at the other pie chart here. So this chart shows like, you know, there is only 0.1% case that the embryos were transferred at day 1, and the rest of them, like 99% of the cases, the embryos had to undergo extended in vitro culture. So at this point, you may say, okay, I see. So what? These strategies were initially taken to improve the successful rate of IVF. That is right? But scientists do not stop here. Let us think, what are the potential problems of these strategies? Like everything has two sides. Now I would like to show you the side effects of these two major strategies. First of all, the multiple embryo transfer. So we can see that it's very straightforward for us to think in this way. Multiple embryo transfer dramatically increases chances of multiple pregnancy, right? So in this chart, the green line here shows the single infant birth rate, and the gray line here shows the multiple pregnancy and multiple babies birth rate in the year of 2013. And you can see that between the green line and the gray line here, there is a big gap, suggesting like more people in the IVF clinics, they tend to have more babies, more than one babies. So the adverse impacts of multiple pregnancy here are many, including increased likelihood of having various medical complications during pregnancy and child delivery. And this can be very dangerous to mom and fetus. Besides medical factors, we also fail to count the emotional cost, the financial cost, timing cost, etc. So in one summary, no matter how, it does not hurt for us to try to be in control here about how many babies our patients are going to have.
Yeah.
Another pressing question that is reflected from the data, as you can see that the outcome of the ART in aged people are extremely low. As you can see, the y-axis is the aging factor. As you go beyond like 40, the chance for you to have a baby is really, really low. I want you to keep this in mind. I'm gonna show you why my research can improve that part too. So it turns out that this is not the only one, we're not the only one who thinks out the problem is a pressing one. So the chart on the right shows that there is an increasing percentage of people who chose to use elective single embryo transfer method during the past 10 years because they want to have a better control about their life, about the quality of their life after conception and baby birth. Okay, so the next question I wanted to show you is about the side effects of extended culture time for embryo. So what are the adverse effects about this procedure? Well, this method cultures embryos, human embryos in vitro for many days until the embryos reach blastocyst stage. It's true. It is a great help for our embryologists to select the best embryos with the best qualities. However, it is well proven by many research that extended embryo culture have adverse impacts on the epigenome of embryos. So nowadays, as the offsprings of ART has become a substantial proportion of the population, the safety of ART has gained increasing attention worldwide. So in the globe, there are many publications reporting increased risk of health problems compared with the natural conceived kids. Among the pathogenetic causes, epigenetic abnormalities are playing a very important role here. Well, most researchers and doctors are paying their attention to the successful rate. I'm confident that the future direction of this technology is to emphasize the quality of life. All right, so in this slides, I'd like to sum up what we have just gone through. Nowadays, the three existing problems of the current ART technologies are, first of all, uncontrolled numbers of babies. Second of all, less than optimal embryo quality and extremely low birth rate in aged populations. Well, you know, there are many ways to attack these problems in scientists' brain. In my opinion, the basic reason that causes these problems is that we do not know which egg is of the best quality that can sustain a full-term embryo development. That is why multiple embryos were transferred. That is why embryos need to be cultured for more than 3 days to make sure they're in a good shape. And that is why aged people have a smaller chance of getting pregnant because the majority of their eggs are not at their prime condition at all. And the doctors do not have an effective way to recognize the eggs from many eggs that are suitable for embryo transfer. That is when the question came to me, can we find a biomarker with which we can find the golden eggs effortless, like this? Can I use Zinc sparks as a biomarker that helps to identify the egg and embryo with the best qualities? And you are gonna be like, hold on, what is Zinc sparks? Right? Okay. So now with the next slides, I'm gonna explain to you what do we know about Zinc sparks. So first of all, I'd like to refresh your memories about gametes biology that you learned at college, and also introduce to you how did we land on the hypothesis that Zinc spark can serve as a biomarker of egg quality. Now let's take a look at the basics of a mouse oocyte, for example. The intrinsic mechanisms of and events that happens in human eggs are very similar to mouse, except that we have different time frame. We all know that in a sexually matured individual, oocytes are always arrested at prophase 1 stage in ovaries. Once the hormonal stimulation kicks in, the oocytes will resume meiosis process and finish the extrusion of first polar body, and as you can see the polar body here, and get arrested at second meiosis stage at metaphase 2 stage. So this is the stage that keep the egg in a fertilizable status. As you can see that a healthy egg at this stage usually had its DNA concentrated here in a spindle form, and stay in the oviduct waiting for sperm to come. So once the egg meets with sperm, the egg will finish the second meiosis within 6 to 8 hours and develop into a 2-cell embryo within 16 to 18 hours post-fertilization. During this important period, previous graduate students and postdocs in Dr. Woodruff and O'Halloran's lab found that the intracellular zinc level changes dramatically between different stages. So specifically speaking, we found that one single oocyte is able to take up 20 billion zinc atoms during the 14 hours maturation window. Mm-hmm. And then it dumps 15 billion zinc atoms during the egg to embryo transition period. To answer, this is very dramatic. To answer why does an egg bother to mobilize so much zinc during the egg development and embryogenesis? So we found that zinc actually can act as a signaling molecule that regulates cell cycle via a protein called Me2. So interestingly, to prove this very physiological nature of zinc signaling, we and the other group at the same time found that simply by manipulating the zinc level, we could artificially activate mouse oocytes. So this method even led to live birth. Here you can see the live birth of this by simply deplete the zinc level, from the intracellular. So this suggests that the decreasing zinc level is essential for the egg activation or egg to embryo transition. In the effort of finding out how does an egg decrease its zinc content in such a short time during fertilization, we found that eggs of mouse and other non-human primates are able to release a large amount of zinc atoms during egg activation via exocytosis. So I would like to play a little video here to give you a rough idea what does a zinc sparks look like here. So this is a 3D animation. As you can see that this event is, as you can see, as you can see that the this event is triggered by calcium oscillations and mainly mediates the zinc loss during activation. So this movie is a 3D animation. Let me play it again. The green dots here, it looks like these are labile zinc stored in millions of vesicles in the cell. And once sperms comes in, this egg will release millions of release a large amount of zinc into the extracellular environment, as you can see the yellowish or reddish light here. So having seen what zinc sparks looks like, let's summarize the characteristics of zinc sparks. So first of all, it is triggered by intracellular calcium transients, as we can see here. So the black line here represents the calcium signals, and the green line here represents the zinc spark signals. It is coordinated by intracellular calcium transients. And then the second trait is it occurs rapidly and extracellularly. This is very important because we know calcium has been well correlated with embryo development in the past 10 years. However, calcium can never be used as a biomarker because its detection is invasive because calcium oscillation happens inside of the cell. However, with zinc sparks happens extracellularly. We kind of hope that zinc sparks pattern can reflect the calcium activities in a cell. And that's one of the reasons we hypothesis zinc sparks can predict embryo development. And the third is zinc sparks can also happen in non-human primates. Therefore, the three aforementioned traits have made zinc sparks an ideal non-invasive biomarker. It is at this point we decided that we need to investigate zinc sparks technology as a quality marker for human medicines. Okay, let me show you the hypothesis there. So we hypothesize that the zinc sparks that is released from the mature egg into the external or extracellular space at the time of fertilization in a series of exocytotic events termed as zinc sparks is an ideal non-invasive biomarker of egg and embryo qualities. Two outstanding questions we wanted to address is, first of all, does zinc sparks happen in human eggs or not? And the second question is, what is the relationship between the zinc sparks and the pre-implantation embryo development and quality? And this study will be conducted in mouse model, as I will show in the slides, because human embryos is not allowed for research purpose. Okay. Now we proceed to the next slide. Oh, sorry. Okay, so this is just. Oh. Okay. So this is the first data I'd like to show you for the first project. First of all, I'd like to show you that we obtained the human eggs from Fertility Center of Illinois. We collected immature female gametes from ICSI patients that would otherwise have been discarded as routine practice. At the Fertility Center of Illinois, which will be briefed as FCI, only eggs that are at the metaphase 2 stage within 2 hours of retrieval are used clinically for insemination, and all other immature eggs will be discarded. And these eggs are going to be the eggs that we are going to use. So from the figure on the top left, you can see that although only 22% of the eggs or gametes arrived at metaphase 2 stage, however, based on our experience, 90% of the immature eggs were able to reach metaphase 2 stage within 24 hours after in vitro maturation. To evaluate the quality of these eggs we obtained from the center, we found that the diameter is within normal range. As you can see here, the diameter is very normal compared to the healthy eggs and the references. And we also checked its immunostaining, and the picture here on the right shows that a typical human metaphase 2 egg, it has a characteristic cytoskeleton morphology, including cortical actin filament, which you can see as a red color here. And it also has a bipolar spindle, which is the green color here, as shown in the immunostaining picture on the right, suggesting that once the eggs arrived at metaphase 2 stage, they are pretty healthy eggs in our hands. So to evaluate the zinc level in human eggs, we collected 6 metaphase 2 eggs from 4 patients or participants and processed them for X-ray fluorescence microscope. So X-ray fluorescence microscope is a very well-established method to quantify and image distribution of trace elements in biological samples. The elemental analysis demonstrated that, as you can see, the iron and copper level and zinc are uniformly distributed throughout the human eggs. However, zinc and iron were significantly more abundant compared to copper. Such high relative iron levels were not observed in mouse eggs. However, we see it in humans may be linked to the use of high iron-containing prenatal vitamins in these populations. The high content of zinc in the human eggs suggests that zinc is playing a vital role in gametes biology in eggs. Oh, sorry again. Okay. Okay, next slide. And therefore, we went ahead to test if zinc sparks happens or not in human eggs. To do this, we activate human eggs with different methods and observe zinc sparks. Because fertilization is not allowed for research purpose. We activate the eggs with parthenogenetic activation and microinjection of sperm factor to activate the eggs. So the movies and figures on the left shows that the addition of 20 micromolar ionomycin, which is a calcium ionophore that is commonly used to activate eggs, immediately induces zinc sparks, as I play the movies here. And you can see that we loaded, in order to see the calcium inside of the cell, we loaded fluo4-Am into eggs. And we used the fluosin-3 dye in the medium to monitor the changes in zinc concentration. And the graphs below the movies shows that the calcium and zinc traces, which were made by image fluorescence quantification, followed by statistical processing, it shows that the red color line is the calcium signal, and the green line here represents the zinc signal. And you can see that it always occurs spontaneously and at the same time. And there is a big bump right after the addition of the ionomycin, suggesting that zinc sparks happens during the parthenogenetic activation. However, like I said, as fertilization is not allowed in human eggs, I also use human PRC zeta cRNA injection, which is thought to be the sperm factor that activate eggs, and I observed zinc sparks in human eggs too. As you can see, after the human PRC zeta cRNA injection, the human egg is able to mount multiple calcium oscillations, and we can see zinc sparks right after one single, the first calcium oscillations. Since we know, the next question is, since we know that zinc sparks happen in human eggs, we still don't know the function of zinc sparks. In order to address this question, I depleted the intracellular zinc by treating the human eggs with 50 millimolar TPEN, which is a specific zinc chelator, for 2 hours. And I found that within 30 minutes, the intracellular zinc is completely depleted by TPEN, where the fluorescence of intracellular zinc staining by fluosin-3 was all gone in the TPEN treatment groups. In comparison with the control eggs, it still showed a bright fluorescence after incubation with 0.5% DMSO for 30 minutes. Because human embryos are not allowed for our research setting, I captured these TPEN-treated eggs before they had time to finish the egg-to-embryo transition and fix them for immunostaining. The results shows that TPEN-treated eggs started to show signs of activation, such as polymerization of microtubules, as you can see the green color here. However, you don't see this polymerization of microtubules in these control eggs that has been cultured with DMSO for more than 2 hours. And also, I also see the decondensation of DNA in these TPEN treated eggs. While comparing to the control eggs, it still has a concentrated spindle-like structure DNA. So all these signs suggest that human eggs can be activated by simply depletion of the intracellular zinc. Okay, so far I have shown you that first of all, zinc is abundant in the human eggs, and extracellular zinc sparks are a hallmark of egg activation and occur coordinately with intracellular calcium transients. The zinc spark and calcium transient profiles are coordinated in mature eggs, and the variability is also observed in zinc sparks and calcium transients. This may indicate differences in underlying egg quality. Like I said before, you know, the human embryos is not allowed to use for research purpose. Therefore, for the second objective, which is to determine the relationship between the zinc sparks and embryo development, we are gonna switch the model from human to mouse. Okay. So the movie on the left shows mouse eggs releasing zinc sparks after activation by calcium ionomycin. Let me play the video, show you here. As you can see, the bright field of the egg, they're aligned here. So the moment after I added the anomycin, you can see a big flashy bright. Here it is. So that's a zinc spark. So based on these embryos, in order to correlate the zinc sparks and embryo development, what I did is, right after the zinc imaging, I cultured these single individual. Let me drag back the movie to a place that where you can see. So after imaging, zinc spark imaging, I just cultured these every single individual embryos in the incubator for 5 days, and then based on these embryos' developmental outcomes, I categorize these embryos into 3 groups. First of all is called non-activated groups, because these eggs did not show any signs of activation. It did not show prenuclear formation. It did not cleavage at all after treatment. The second group is called non-blastocyst embryos. So these embryos were successfully activated. However, they either arrested at a certain stage or underwent apoptosis before reaching blastocyst stage. So the last group, the third group, is called blastocyst group. So these eggs were able to be activated and they were able to reach blastocyst stage after 5 days culture. Using software ImageJ, I can analyze the amplitude and the total release of the zinc sparks of each egg. And the image on the right is the maximum projection of the movie. And the fates of these embryos were noted. And you can have a very subjective impression that those eggs which release brighter zinc sparks have a better developmental results compared to those eggs with fainter zinc sparks. However, these eggs who did not release zinc sparks or release very, very faint zinc sparks were not activated at all. And it turns out that the statistics further confirms the information that is conveyed by the previous image. What I found is those unactivated eggs, which are denoted by the black color here, displayed the lowest zinc spark intensity. And the blastocyst embryos release a much stronger zinc sparks in terms of amplitude and the total zinc release compared to the non-blastocyst embryos and the non-activated eggs. However, we may keep in mind that these methods were performed under parthenogenetic activation environment. It's not physiological experiment. In order to confirm what I found, I performed IVF and imaged zinc sparks during this process. So in these slides, I want to show you a real-time movie that demonstrates for the first time zinc sparks occurs right after sperm enters the enters the egg. So if I click the movie here, if you pay attention to, sorry, it's not playing. All right, if you pay attention to this little guy here, which is the sperm, and as time goes by, the sperm goes in, and the egg releases zinc sparks. And this is the first time, like I said, this is the first time in human history that we were able to capture the zinc sparks right at the moment of sperm entry. And this is something big to prove the physiological nature of zinc sparks. Using the same analysis process as I just showed you, I found that the blastocyst embryos release much more zinc during fertilization than the embryos that fail to develop to blastocyst. As you can see here, the red color here represents the blastocyst embryos, and the blue color represents the non-blastocyst embryos. And it is much more the zinc amplitude and the total and the rate of release is much higher compared to the non-blastocyst embryos. Okay, so far the data suggests that the pattern of zinc sparks is closely related to embryo development, but whether zinc spark can also predict the quality of embryos is still not known, right? So this is an important question because in human medicine, day 3 to day 5 embryo transfer is the most popular method Therefore, being able to predict the quality of the blastocyst at day 3 to day 5 is really a big deal here. To address this question, what I did is I counted the cell number of the blastocyst embryos after zinc imaging. So the total number of the blastocyst embryos is a golden standard to reflect the healthy status of blastocyst embryos. Interestingly, I found that the zinc spark amplitude is significantly correlated with the total cell number of blastocyst embryos. And the R value is, I'm sorry, maybe here is here. The R value here is almost close to 1, suggesting that the correlation between these two factors are very, very strong. So this data suggests that simply just by looking at the zinc spark at the time of fertilization, we can not only predict which embryos can develop into blastocysts, but also we can predict how the quality of the embryos will be 5 days later. So the conclusion for the second objective is using parthenogenetic activation and IVF methods, we found that embryos with better developmental potential release zinc sparks with higher amplitude and greater total release compared to the embryos with suboptimal quality, which includes unactivated eggs and non-blastocysts. So this data, in summary, supports our hypothesis that zinc spark is an extracellular hallmark of egg activation and can be effectively correlated to embryo development.
Interesting.
Yep. Okay, next slide. So the future direction of our research is to evaluate the predictive power of this zinc spark technology on pregnancy and live birth in mouse. And we also want to address, can we improve IVF outcomes in aged eggs using zinc spark technology? And plus, we also want to apply what we have learned from mouse to human beings. And to do this, we're going to determine the relationship between the zinc spark profile and egg quality in human eggs. So at the last slides, I would like to thank Dr. Woodruff and Dr. O'Halloran, who have initiated this grant and project at the first place and have been supervising the development of this grant along the way. And this project won't be possible without their supervision and wisdoms here. And the co-authors of this study also contribute significantly to this grant. Dr. Francesca Duncan and Emily, as I show here, their name is here, are the two co-founders of the Ferring grant. They did a lot of work to make sure that we got human eggs from FCI with good qualities, and everything else about this project were on the right track. And Dr. Eve Feinberg graciously provided human eggs and samples to us, and she also provided precious insights helping us to envision this study in the eyes of a clinician. Also, I would like to thank all the lab members of the Woodruff lab and O'Halloran's lab. This is really collaborative work that is not possible without their help and support. And this funding was supported by the Thomas J. Watkins Endowment and a research grant from Ferring Pharmaceuticals. And finally, thank you all for your time and attention. And I'm happy to take any questions.
Thank you, Dr. Zhang. So if you can unshare the presentation.
Okay.
Great.
Okay.
I have 1 or 2 questions.
Sure.
One of them is like you are using X-ray to monitor the zinc levels.
Mm-hmm.
Does it have any effect when you implant it to the, like, since you are exposing the embryo to the this thing? So does it have any impact on this one? Okay. The eventual birth of this one, the pups and all?
Okay, that's a great idea that I'm glad that you bring this up, because the data I'm showing here just to give you a rough idea about the two papers that I published. If you go to the paper and take a look at the data in details, there are other figures to show the total number of the embryos have been greatly compromised by the current zinc imaging condition.
Okay.
This is kind of expected, because we are putting a high concentration of zinc dye in the medium, and we are not sure about the side effects of this dye, and plus these eggs have been undergoing intensive exposure to laser bombardment during the imaging period, which is not good for the health of the eggs. And obviously right now the technology or the imaging condition here is not invasive at all. So that's also another potential future direction that we're going to pursue with, is to develop a greener methods, whereas we can see the zinc sparks. However, these eggs will not be exposed to any dye, will not be exposed to any laser exposure, and that's gonna be one of the major hurdle we're going to overcome in the near future, in order to make this technology applicable to human medicines. Great questions.
And I have another one. Like you initially mentioned about the epigenetics, right?
Yes.
Like when you have this, like there might be epigenetic change.
Mm-hmm.
So have you done these experiments, like after implanting, and went through the epigenetics profile of the mouse pups or anything like that?
That's an excellent question that you just asked about, because we see, based on the previous literature, we saw very, very close correlation between the ART and the health status of the offsprings. And like you said, epigenetic modifications or abnormalities is a major concerns here. So we are very interested into looking at, if we can predict the epigenetic abnormalities using zinc spark technologies. And that's also another future direction that we're going to pursue. But that will happen for sure after we figure out how to detect or image zinc sparks during the fertilization without harming the eggs. So beautiful, excellent questions. These are our future directions.
Can you measure the zinc levels using MS or something like that in the media where you culture these embryos?
Well,
Or
now
like
I start to kind of wish that I have you in the team, because you are thinking exactly like what we do in the lab. So how do we tackle down these questions? Measuring the total zinc in the medium will be a way to go. In this way, we can just let the eggs and the sperm do the fertilization part without adding anything in the medium, like what we usually do in the lab. So after the fertilization, we can just take the eggs out and measure the medium and see what's going on there. So yeah, but there are a lot of technical problems that is going on here, because considering that the small amount of zinc that is released by a single one egg, so we need to find some sensitive method to test this. We also have other great ideas and opinions going on here about this project, but that's a very brilliant idea, I have to say.
Okay. Yeah, like it's really an interesting project. I think you will overcome all these technical hurdles, and in someday I think we will be able to see some of the good work in the hands of patients, where we will be able to evaluate the eggs okay, only one egg is, this is a good egg where you can implant it. And so thank you so much. I really appreciate you taking time to come to our science hangout and present your work. Really, I think everyone will enjoy your presentation. Thank you so much.
It's my pleasure. The pleasure is mine. Thank you so much for your time.